This is a short video on fungi and antifungal agents. We're going to start off by talking about a couple unique features of the fungal membrane and how antifungal agents target those features and hopefully eliminate a fungal infection without hurting mammalian cells too much. So here on the left, we have a picture of a fungal membrane, and we're going to talk about some defining features. The top, we have the glycoproteins, and these contain mannose. Now, we don't have an agent that's specific for mannose, but we do know that mannose is one of the triggers to the complement pathway. So this is what enables our immune system to help clear the fungi. We also have some beta-glucans, specifically beta-1,3 glucans and beta-1,6 glucans. And the 1,3 and 1,6 just talk about the kind of linkage that's involved in those polymer chains. We have chitin, which is another polymer. And inside the phospholipid bilayer, we have ergosterol, which is kind of an analog to cholesterol, which is found in human cells, of course. Ergosterol plays a similar role. It's a steroid. It helps regulate the fluidity of the membrane. And just to note at the bottom left here, we have DNA synthesis that's going on inside the fungal cell, and there will be an antifungal agent that acts against that. So let's talk about the antifungal agents. First, we have echinocandins. There's a few listed here. They all end in fungin, and they particularly inhibit the enzyme that synthesizes beta-glucans. So essentially, these are called the penicillin of antifungals because penicillin worked to inhibit the cell wall formation in bacteria, whereas echinocandins work to inhibit the formation of beta-glucans, which make the cell wall of fungi. Next, we have polyenes, such as amphotericin B. That's the biggest, most commonly used one. It's a polyene, and these bind ergosterol, and they essentially weaken the membrane, cause a big pore to form in the membrane, which causes some ions to leak out and essentially kill the fungal cell. One of the problems with polyenes is that they sometimes can also bind to cholesterol, and this can lead to some mammalian toxicity as well. But polyenes, such as amphotericin B, bind ergosterol. We also have azoles, and these are easy to remember because, as you can see with the three examples here, they always end in azole. They inhibit the synthesis of ergosterol. So polyenes inhibit the activity of ergosterol, whereas azoles inhibit the synthesis of ergosterol. And lastly, we have 5-flucytosine, 5-FC, which is converted to 5-FU. Fluorouracil, which you might be familiar with, is a pyrimidine analog that inhibits DNA synthesis. One of the side effects of 5-FU is myelosuppression. So this happens when you treat a fungal infection with 5-flucytosine. And real quick, let's review them one more time. Echinocandins block beta-glucan formation, beta-glucan synthesis. Polyenes block ergosterol activity. Azoles also block ergosterol. In this case, they block ergosterol synthesis. And 5-FC, which gets converted to 5-FU, inhibits DNA synthesis inside the cell. Now we're going to talk about a few categories of the fungi. We start with the yeasts. Yeasts are unicellular organisms, and two big ones that are important to know are Candida and Cryptococcus. Next, we have molds. Molds are multicellular organisms. They can get big enough so that they are macroscopic. You can see them without a microscope. We have Aspergillus, Zygomycetes, and Fusarium. Then we have dimorphics. Dimorphics can switch between being a yeast and a mold. Usually at body temperatures, 37 degrees or so, they are yeasts. But at room temperature, maybe 20, 25 degrees, they are molds. There are four that are important to remember: histoplasma, blastomyces, coccidioides, and paracoccidioides. Now we're going to talk about how the drugs that we talked about, the four antifungal agents, inhibit yeasts, molds, and dimorphics, and kind of their range. This is just a brief overview of the range of action of these antifungal agents. So echinocandins, again, the ones that inhibit beta-glucan synthesis, they work in the Candida and Aspergillus genuses, and they do not work on the dimorphic fungi. The polyenes, which, as we said, inhibit ergosterol activities, they work in all of these listed here except for this one Aspergillus species. Azoles, which inhibit ergosterol synthesis, work in all of these listed here except for Candida glabrata. And they also have, they're slightly ineffective. One of the azoles is slightly ineffective in one Candida species and in the molds in general. But in general, azoles are usually pretty good against all of these. And then, of course, 5-FC works in the yeasts. They do cause some resistance in the yeast, so it might not be your first choice. And it does not work in the molds and dimorphics. So of these shown here, it seems like polyenes have the broadest range, whereas 5-FU or 5-FC that gets converted to 5-FU has one of the smaller ranges. This has been an overview of fungi and antifungal agents. Hope it helped. Thanks for listening.